Hello and welcome to the nanomedicine lecture on targeting. So first of all, we're going to talk about active versus passive targeting and targeting in general. So why would you ever want to target something? We've already talked about in class about the enhanced permeability and retention, or the EPR effect, where tumors or other things can accumulate nanoparticles just passively there into the tumor. And so why would you actually want to target, and what is targeting? So targeting is putting a ligand onto a nanoparticle that will specify the nanoparticle to go one place or the other. So this allows you to increase specificity of the nanoparticle. So in that way you can get tumor versus normal, or you can get one cell type versus the other cell type. Now mind you, it's not binary, it's not on or off. You can just change the distribution by a little bit, and I'll show you examples of this later in the lecture. With targeting, you can change biodistribution. Again, with nanoparticles, you cannot get rid of the kidney, the liver, spleen, the organs of the RES, but you can distribute them more into the tumor, for example, than into the liver and spleen, and even greater than in the passive targeting of the EPR effect. You can increase the efficacy to toxicity ratio, and so you can use much less drug in a targeted nanoparticle than you would use in a free drug, and hopefully that would decrease the side effects, the toxicity. And then finally, active targeting is a myth. So putting a ligand on something does not mean that that nanoparticle will home only to the site that you want to target. That's not how it works. It still does the pharmacokinetics. It still will, if it's an intravenous injection, it will still distribute homogeneously throughout the whole bloodstream and then be taken up past the blood vessels at the same rate. It's just that the targeting ligand will bind to whatever molecule that you've designed it to bind to, and there it will stay and will accumulate. So it's not active targeting. It's not a T cell or any kind of white blood cell where they can chemosense and they can go and seek out things based on a gradient, a chemical gradient. That's not what happens here. So active targeting is a myth. It's both passive targeting. It's just when it gets into the tissue, it binds there and is taken up by cells, or it binds to that molecule on the cell surface and releases its drug there. And that allows it to stay and accumulate. And it's less prone to clearance. So points to consider when you are targeting. What is the application? Do you need nano at all? If it's a small molecule and you need it to be an acute effect, like unblocking a clot in the heart or a stroke, you wouldn't want nanoparticles. We know from our biodistribution lectures and our distribution lectures that in pharmacokinetics, that the blood half-life of a nanoparticle is very long. So that's not a good idea. It's not a good application. And then with the do you need to target, so if you want to target macrophages, you don't need to do anything fancy. Most of the nanoparticles will be taken up by macrophages or phagocytic cells quite well. So if you want to target a macrophage, no, you don't really want to do that. When would you need to target? Well, I'll show you an example in the heart, but you would want to, after a heart attack, cause the cardiomyocytes to proliferate, so that way you could get more cardiomyocytes and less fibroblasts. You do not want that drug that would cause proliferation everywhere throughout the body. You want it only in the cardiomyocytes, and so then you need to target it directly to the cardiomyocytes. And then finally, what you need to consider is where do you need to deliver the molecule? And again, this is all about choosing the appropriate target. So what is the appropriate target? The appropriate target should be of high abundance. The more receptors, or the more the target is, the better this is. And you'll see this in a little bit when we talk about Bmax and pharmacodynamics. It should be selective. If you want it to target a tumor, for example, and not the normal, you do not want your target to be present on the normal. It should be only present on the tumor. It should be accessible. We talked about the barriers. You do not want the barrier, you do not want it in the nucleus, for example, because then you would have to go across multiple different barriers. So you want it to be accessible. And you also want to be able to make a targeting ligand against it. Not everything can you make a targeting ligand against. So if there's not something that will bring it or cause it to home directly to that target, that won't be a good target. You can't do drug delivery or imaging to it. So target selection is key, and target selection is the critical parameter, one of the critical parameters of importance. If you get the wrong target, no matter what you do from now on, you will not have a successful drug delivery program. So there are many classes of targeting ligands, and they have to be high target affinity. They have to have high target to background ratio. So again, that's the selectivity. And you want them to escape the macrophages. You do not want them to go into the macrophages. And how this is done is antibodies. There are antibodies out there to almost every single protein. But antibodies have advantages where they have really high affinity and the structures are well known. However, you have to know what you're targeting with them, and they can be immunogenic, and they are difficult to produce. And antibodies have to be humanized, and since they have to do this in a cell-like experiment, they are classified as biologicals by the FDA, and so that's another whole regulatory process. Another way to do this is peptides, and this is what my lab mostly focuses on. These are small amino acid chains, 7 to 12 amino acids. They're non-immunogenic. They can be made against anything, even if you don't know what the target is and you want to identify it later. But they have poor pharmacokinetics, as we've seen. And sometimes in the selection procedures, their affinity isn't high enough, and affinity is a really important parameter. Aptamers are DNA or RNA molecules. They're not used very often because in the bloodstream, the nucleases degrade them very rapidly. And to cause that not to happen, you have to use a fluorine or a methoxy group on the sugar in one of the positions in the sugar. And those are not really easy to make, and there's potential for toxicity. So aptamers really aren't used that often. And then organic or diversity-oriented synthesis molecules. These are small molecules that you can use as targeting moieties. So they're not drugs themselves, or they're not typically drugs themselves, but you can conjugate them. You can conjugate fluorophores or radioactive nuclides to them. Radioactive nuclides would be for imaging and for drug therapy. Or you can also conjugate them to nanoparticles, and they will target the nanoparticles. Really no disadvantages to organic or DOS molecules, except for they are much more difficult to screen for binding pairs. For them to bind to the target you're interested in, there's no way to deconvolute them, so drug companies have multi-million-dollar pieces of equipment in order to do this high throughput screening, and they also need a bioinformatics suite of tools to do this, so that's their disadvantage. They're not easy, and it's certainly not for the faint of heart to do. When we are making a targeted ligand, you absolutely have to consider affinity and Bmax. Bmax is an easy parameter. Bmax is the number of receptors per cell, or the number of binding partners per cell. This is this dotted line up here. That is the Bmax. Affinity, on the other hand, is the concentration where half of the receptors are full, so half Bmax. That's the affinity. That's called KD. That is called KD, or the affinity constant. And here's examples where you can have different Bmaxes, or you can have different EC50s or KDs in this case, where they have the same Bmax, or the same number of receptors, which it has to be. So if you are binding to the same receptor, the same site on the receptor, No matter what ligand you have, the Bmax is the same because the number of receptors per cell or the number of binding sites per cell does not change if it's the same target. So the target level does not change. However, the affinity of the ligands can change. So you can make a high affinity ligand, which is this, or a lower affinity ligand, and that's where this KD parameter, this 50% binding, changes. There's also something else called cooperative binding, or the Hill equation. So this is the fraction of the molecule saturated by ligand as a function of the ligand concentration. It determines the degree of cooperativity of the ligand binding to the enzyme or receptor. And so what happens is as you get one ligand binding to the receptor or one targeting agent to the target, it changes the conformation of that target and makes a second or a third target easier to bind. Or if you have an aggregate of the targeting ligand, it binds better than the original. So a coefficient of 1 indicates completely independent binding. So no matter how many ligands are bound, you don't get any effect in the affinities to change that. Numbers greater than 1 on the slope will indicate positive cooperativity. Numbers less than 1 are negative cooperativity. So positive cooperativity is that after one ligand bound, the second and third and fourth or fifth, depending on how many sites there are easier and easier to bind. Hemoglobin is a good example of this. It has a Hill coefficient of 2.8 to 3. Negative cooperativity means that once one is bound, it is very difficult to get a second ligand to bind. And here's an example of what these curves look like. So these are sigmoidal dose response curves. This is total binding on the y-axis versus concentration, or the log concentration of the drug. And this is an inhibition curve because this is log unlabeled drug. If this was a binding curve, the sigmoid would start low and then go high and reach its Bmax. So this is still Bmax, but this, instead of KD, is IC50, which is inhibitory concentration 50. So this is the concentration where 50% of the binding is no longer present. So you've inhibited it from binding. These are 4 parameter curves where the bottom is here, the top is the Bmax, and this slope is the Hill coefficient. And there's a 4th parameter, which is this IC50. So those are your 4 parameters: top, bottom, Hill slope, and the IC50, or affinity, KD. And here are the equations to be able to understand this. Here's the derivations for the Hill equations. Here's the competition curves. You can also linearize this, where this is A is the Hill slope of 1. That indicates no cooperativity, or they're independent. B is negative cooperativity, and C is positive cooperativity. So now let's switch over from binding to pharmacodynamics. But before I do that, just one more point about point about binding. Affinity is very, very important. The higher the affinity, the more tightly it will bind. You also need very high numbers of Bmax. The more receptors per cell, the better that is. So pharmacodynamics, efficacy and potency. There is a difference between potency and efficacy. And so potency looks like Bmax from binding, but this is response. This is not just binding. This is the drug is actually eliciting a response. So if you want the cells to die, this efficacy is the max number of cells that are dead. Potency, on the other hand, is the amount of drug needed to produce a given effect at 50% of the Bmax. So that's potency. It looks like KD, or affinity, but it's not. It's, again, based on response. Curves are exactly the same, mathematically. However, the Bmax is efficacy for pharmacodynamics, and the Kd or the affinity is potency. Drugs can have different efficacies and they can have different potencies. And then finally here there's therapeutic window. So with the therapeutic window on the axis, the y-axis, again, another very important parameter. This is determined in terms of plasma concentration. This is not tissue concentration. Remember, it's very difficult to quantitate tissue concentration, so we do this by plasma. This is why plasma is always the first compartment, and it's always incredibly important to know the concentration in the plasma. So here's the therapeutic window graphically. This is plasma concentration levels over time. Here's the curve. You should be able to recognize this as a one compartment model curve with absorption. Here's your duration of action. And that's in between, this is where the curves cross or bisect the in the desired response from the subtherapeutic. So down here, the yellow is the subtherapeutic. So in that concentration range, there is no action. The green is your therapeutic window. So that's the area where your desired response is. And then after that are side effects or adverse responses. So here in the reddish pink is the danger zone. That's the toxicity. And so the duration of action is where it crosses the desired response and subtherapeutic response. If a drug gets up into the side effect range, then you will have an adverse reaction. Most drugs are tailored, or all drugs are tailored, so that way the concentration in the plasma maintains peak effect or close to peak effect without going back down to subtherapeutic. But also you can't just inject or take these drugs at whatever time frame, because if you would then add more drug, for example here, instead of letting it clear out to here, then when you dose this patient again, it would go up into the side effect instead. So what you want to do with multiple doses is to be able to keep this and maintain it in the therapeutic window. So here's an example of antibodies. We can fluorescently label them. We could put them on nanoparticles. This, in this example, is a magnetic nanoparticle. And this is an antibody targeted to vascular cellular adhesion molecule. Very important molecule in atherosclerosis, which is a leading cause of blood clots and therefore a leading cause of stroke and heart attack. And here what is happening is we're being able to image the endothelial cells. These are live animals, in animals, the endothelial cells that are activated with VCAM. I told you that aptamers aren't really used, but I wanted to give you some references where this group has been able to do this. They have used RNA aptamers against a prostate serum membrane antigen, PSMA aptamer. And this is supposed to allow drug targeting to prostate cancer. And so here's the drug inside. This is a PLA nanoparticle, so a polymer nanoparticle. Here's PEG is the blue, and then it's a carboxylic acid on the end. They then used an amine group on the nanoparticle, or an amine group on the aptamer, conjugated it, and then from there we're able to deliver drug, which we could see is red, into these tumor cells. They didn't do this, at least in these papers, in vivo, but still you can use these aptamers. We can also look at molecular changes in insulitis. This is just looking at blood vessels, and this is perfusion. And then going into the small molecule conjugated. So these are those little small molecules, the organic or DOS molecules. And so if you have a nanoparticle that has, this is again a magnetic nanoparticle. These are coated with amines, so you can functionalize them. And then using different chemistry, EDC or NHS, you can go to amines. You can use this molecule for anhydrides, SIA or SOCl2 for hydroxyls, Again, EDC NHS for carboxyls, SPDP for thiols, or you can use epoxides themselves. They will react with these amine nanoparticles. That way you can build a library that has, each nanoparticle has a different small molecule. Again, this is not an easy task to do. We synthesized roughly 386 of these and then did cell uptake and quantitative assays to build a heat map where we could see exactly where the different particles are binding. So green means it's high binding, black means it's average, and red means it's lower binding. And so here's a cluster here that is binding highly to these cells, but not binding to the endothelial cells. And so in this way, you can try to make a library or screen a library for small molecules that will bind. From here, we were able to look at this in endothelial cells. So there are these 4 compounds that bind really well to different endothelial cells. And then from there, because they're magnetic and fluorescent, these nanoparticles, we can actually image them being taken up by the endothelial cells and the parasites very specifically. We can also look at macrophages. So I told you that macrophages are not tricky to hit. That is incredibly true. Macrophages will take up these cells. However, there are different subsets of macrophages. There's M1 macrophages, which are inflammatory macrophages, and there are M2 macrophages. And in this case, M2 macrophages are also called tumor-associated macrophages, or TAMs here, TAMs. And you would want to image those specifically over imaging all subsets of the macrophages. And so we were able to use one of those small molecules from that screen, putting them on a magnetic nanoparticle, and they were able to direct it to the myeloid-derived cells, and in this case, these are the tumor-associated macrophages. This is what this looks like in vivo, and we can actually see the green is a tumor cell. The red is the macrophages that have taken up the targeted nanoparticle, and over time we can watch the macrophages bind and touch the cancer cell. And so we can start to understand and quantitate the interactions between the tumor cells and the macrophages. And this is very important because the tumor cells are surviving and persisting even with an immune system. So the immune system something is misfunctioning in the immune system, that they are able to bind to the tumor cells, but they're not recognizing the tumor cells as harmful. And instead they're letting them go on their own and continue to do what they're doing. And so a lot of effort has been done in research laboratories, in academia, but also in pharmaceutical companies in order to be able to understand why that immune system is malfunctioning and to develop drugs that will stop it from malfunctioning and instead get it to recognize the tumor cells as foreign or as bad. This is ex vivo imaging where you can deplete the tumor from macrophages by using claudinate. So this is just showing we can quantitate how many macrophages are in that whole tumor. And so in that way we can not only monitor the individual interactions, but we can also very quickly quantitate how many of those macrophages are in there. So if you're trying to get more macrophages or you're trying to get less, we can quantitate that, and so we can really watch how our drug is working. So moving on from small molecules and antibodies and aptamers, we'll talk a lot in this next part about amino acids and peptides. So peptides and amino acids, it's biology, but it's really some exquisite chemistry. There's all sorts of side chains on here where you have just regular alkanes, but then you also have amino acids here. You have amines, glutamine, asparagine. You have the amino terminus. You have carboxylic acids. You have positively charged groups. and you have aromatic groups. So there's a lot of organic chemistry, and a lot of the binding is done through protein-protein interaction. So we've been able to harness the power of these amino acids to make peptides that allow us to find specific targeting ligands. When two amino acids bind, they form an when two amino acids bind, they form a peptide link. So this is a dipeptide. Here's the peptide link, and then it releases water. Because of their side chains and the way the bond is made, they're a steric hindrance, so there's only so many angles these can assume. So the phi and psi angles are very important for this, and you can only have so many phi and psi angles, and this translates to secondary structure, where you have a beta-pleated sheet. These are these angles, an alpha helix or a left-handed helix. That's all that can happen. And so what we've done, not me, but George Smith from the University of Columbia, at University of Missouri at Columbia, he is a virologist, and he hypothesized that he could display peptides or antibodies on the surface of viruses or bacteriophage, and bacteriophage are viruses that bind only to bacteria, so they will not infect and replicate inside of a mammalian cell. Only bacteria, and only in their specific bacteria. And these are really interesting molecules, and we can engineer them because they're very simple. There's only 9 proteins, and because of that, virologists have been able to study them. We know what they do. So the P3 coat protein binds to the F. pilus. That's the male part of the E. coli. It then injects its DNA inside. This phage has single-stranded DNA. Once it gets inside, the bacterial cell will start making it into double-stranded DNA. And this is in equilibrium because enough single-stranded molecules have to be around to package into more phage because it wants to reproduce and replicate. However, you need the proteins to make the coat of the capsid of the phage, and in order to do that, the bacteria has to recognize it as double-stranded. So the first part is it gets it to the bacteria to make double-stranded DNA. From there, you get gene expression, which makes all the coat proteins, and then also makes this P5 protein that keeps some of these single-stranded molecules as single-stranded molecules, and then starts initiating the packaging into the new viruses. And what George did and was able to do was to take the P3 coat protein and then also the P8, which is here, and then display peptides. So the P3 has peptides here, or the P8 has peptides here. And in this way you can screen, and genotype equals phenotype. Because you've specified this inside the DNA of this phage, you can throw billions of different peptide sequences being put on the phage onto cells, allow them to bind, wash them off, and anything that sticks you then go and sequence, then you know exactly what was binding. And those peptides represent a targeting moiety. It's a peptide. So engineering this, we can make them, they're native, so these are nanowires. They're 10 nanometers wide by 1,000 nanometers long. You can make shorter versions where they're 10 nanometers wide by 50 nanometers. Here are the plasmids. You don't really have to go into this, but here's either the P3 or the P8. And then you can do some interesting things with them. You can make them all sorts of interesting wires. So if you display peptides that will, for example, bind gold or iron oxide, you can get them to make a nanowire. And you can also get them to target. So this is one particular virus that I've made that has iron oxide for MR and for heating for therapy, targeted to a protein that's really important in metastatic tumors. How do we make a phage display library? Well, I don't, but New England Biolabs does. And this again illustrates genotype equals phenotype. This is the P3 leader sequence, so this is the P3 protein, coat protein, and this with the NNM or NNK is the random peptide. So one phage gets one sequence that encodes for one peptide sequence. And so that way, when you go back and you sequence them, you know what phage were present just by being able to translate using standard Watson-Crick base pairing what exactly was the peptide sequence that was doing the binding. So let's go through some real life examples just of the peptides and the phage display. We're going to do this for myocardial infarction and also for pancreatic cancer. So with coronary heart disease, it's the largest killer in the U.S. and other developed countries. Starts with plaque that blocks the arteries. The plaques can rupture, creating a thrombosis, which then causes the plaques, when they rupture, they clot, and then they go to the coronary artery, and if the clot is big enough, it will occlude or block the coronary artery. And wherever the coronary artery is occluded, that tissue is not getting oxygen and nutrients, so that tissue becomes hypoxic and starts to die. And that's the myocardial infarction. If the person is lucky, when the paramedics get there, they can unblock it or keep them alive long enough for them to go into the cath lab, where the clinician will be able to unblock it, put a stent in there, and reestablish flow. But with all that being said, there's still a whole bunch of cells that have died, and so what has to happen is there has to be left ventricular remodeling. And so the cardiomyocytes have all died. You can't just have a hole there. So fibroblasts come in, they form a scar, but that scar isn't a cardiomyocyte, and so it doesn't respond to the pacemaker cells, and it doesn't beat. And so then from there, the left ventricle will remodel, and it will get bigger. It won't pump as well. It'll be much more difficult for it to pump, and this can lead to heart failure depending on the size of the LV dysfunction. And so it's really important to be able to do tissue engineering in order to stop that from happening. And so people have used small molecules or proteins to various successes, cell delivery like stem cells, tissue engineering where people are trying to take chunks of tissue and place it back into the heart, or what I'm going to show you is reprogramming the non-myocytes to cardiomyocytes. And we can do this, or what my lab has been interested in collaboration with AstraZeneca, is to cause remodeling of that infarct. So again, you guys have seen liposomes. You know that they're a bilayer. The drug is on the inside. We've made them so they're also visible, so we can keep track of them through our imaging equipment. And then the peptides. So we have done phage display screening to identify peptides specific for the cardiomyocytes, cardiac progenitor cells, endothelial cells that are activated, not endothelium throughout the whole body. You would not want something targeted to the endothelium throughout the whole body. You only want it to the infarct. So we actually have selected peptides specific for the endothelial cells in the diseased heart or the fibroblasts. And we are thinking about delivering anti-apoptotic and proliferative. Proliferative plus differentiating, this would be to the fibroblasts, this would be to the cardiomyocytes. Pro-angiogenic, this would be to the endothelial cells so we can get more endothelium. And then transdifferentiation factors, delivering that to the fibroblasts to get them to become cardiomyocytes. And to do this, we had to use a mouse model. And we chose 4 days after the infarct because this is on the transition between the inflammatory, these are the M1 macrophages, they are remodeling, to the reparative M2 macrophages. So we wanted to hit it right when it's starting to repair. We did an in vivo phage screen, so this is the mouse model we used, suturing this artery in order to simulate a heart attack, and then 40 minutes after, clipping that or reopening that suture so that way it simulates a heart attack that someone had for 40 minutes. 4 days later, we inject the phage that are displaying all the different peptides, tail vein, 4 days. We then euthanize the animal and then grab phage from the different regions, the infarct and the remote. We do not want the remote, we really want the infarct. We then amplify the phage from these regions and then repeat this process 3 rounds. We then sequence them and then look for specificity. And from there, we were able to identify peptides that are specific for cardiomyocytes, endothelial cells, fibroblasts, and cardiac progenitor cells. So now we have peptides that will bind to each of these different cell types. We then chemically synthesize the peptides and we can conjugate them to liposomes. We can also use the phage themselves as imaging agents, and we put fluorescent dye there, and then this is how we look at some of the specificity. So this B40 is an endothelial binder. It's binding to CD31. It co-localizes with CD31, which is an endothelial cell marker. B29 is binding to alpha smooth muscle actin, which is a measure, that's the fibroblasts. B11 is binding to the extracellular matrix. I1 is binding to the cardiomyocytes, this is blue, and then B50 is binding to the c-KIT positive progenitor cells. If you're really interested in this, this has been published in Journal of Controlled Release, and I can get you the paper. I will put the paper up on CoLab so you can see all the details. From there, we conjugated the peptide onto this liposome, and then looking at the pharmacokinetics, we can look and see how much of this liposome is delivered, and we know how much was delivered, and so from there we can calculate the area under the curve. So with the cardiomyocyte targeted peptide, we get twofold more uptake and accumulation into the infarct than if we just use a liposome alone. So this is a great example of why to target. We can deliver it specifically to the cardiomyocytes or to the endothelium or to the c-KIT progenitor cells or to the fibroblasts. Now the interesting part is that there's less endothelial cells and less c-KIT positive progenitor cells than the cardiomyocytes that are in this system, and so the target is more abundant, so you get more there. This isn't saying that this isn't specific for this. This liposome is. It's specific for the c-KIT positive cells, but there's so little of its target there, you're not seeing it above just negative control liposome. And then again, we check for target specificity, and so here's the negative control. This is just the liposome by itself. Like I said, it goes into the macrophages. I1 goes to the cardiomyocytes. B40 goes to the endothelial cells. That's this overlap. And B50 goes to the c-KIT positive cells. And this is the correlation between them. So again, by putting the peptide on there, we are changing the subcellular localization for the liposomes. So here again, this is the correlation. Negative control, or the liposome by itself, goes to the macrophages. That's this blue bar. I1 goes mostly to the cardiomyocytes. But what I want to show you that it's not binary, it's not all or nothing. We were able to shift it by 5 to 10-fold from the other cell types, but it's never negative in the other cell types. There'll always be some kind of overlap with it. It's just we can shift it just a little bit, 5 to 10-fold, which is a lot, but it's not everything. You'll still get macrophage uptake. You'll still get other cell uptake. And then from there, we looked at pharmacodynamics. And so we used a drug from AstraZeneca that inhibits apoptosis or cell death. And so here, this is the free drug. And you're looking for the absence of the green in these cardiomyocytes. So the free drug alone, you still get the presence of the protein that we want to inhibit. Negative control liposomes, you still get that present, but it's no longer in the macrophages. So these are the macrophages out here. And then here's the cardiomyocyte targeted liposomes, and you can see that it's no longer in the cardiomyocytes. And then this is just quantitated. So again, we're shifting this 9-fold from just the drug in the negative control liposomes, just plain liposomes. And we're delivering even more to the cardiomyocytes and getting better cardiomyocyte than free drug alone. And of course, this is the background for the empty liposome. So really, really exciting data in the cardiomyocytes. So that's exactly why to target. We want to be able to get more into the area that we're interested in without it going anywhere else, and we want to deliver it specifically to the cell that we're interested in. And then finally, our last example is pancreatic cancer. So shifting away from the infarct, we're going to pancreatic cancer. And in this case, pancreatic cancer is in the 10 for highest incidence. So that's the number of cases per 100,000 people. However, its estimated deaths, it's around 6% of the cases due to cancer, which makes it in the top 4, and it's projected by 2030 to be second only behind lung and bronchus for the top killer from cancer deaths. And so putting this another way, percent death to incidence, 94% of people that are diagnosed with pancreatic cancer will die within 5 years. The majority of that is between 3 to 6 months. So we really need new strategies. In this case, again, we can't, because pancreatic cancer is diagnosed so late, it's usually metastatic. We had to use a mouse modeling strategy where these animals get pancreatic cancer in the pancreas in order to get tissue that corresponded to early cancer. Once any cancer becomes metastatic, it is very, very, very difficult to cure a patient. Most 5-year survival rates for metastatic cancers are 5 to 10%. So we really want to catch this early. And so from there, we took tumors from these animals, developed them into cell lines, and then we're able to do phage display screening where we incubate the cells, the tumor cells, with the phage, allow them to bind, wash away everything that didn't bind, lyse the cells to recover things that have gone into the cell, receptor-mediated uptake, and then do this process 3 to 4 times, and then sequence to identify the peptides. We can label the phage, or I'll show you later where we can label the nanoparticles, and then look and see the distribution of this inside the tumor with a living mouse using confocal microscopy. But first we took those peptides, we conjugated the specific ones onto the magnetic nanoparticles. They're both MR visible and fluorescent. So we took our peptide hit, conjugated it to the nanoparticle, did in vitro validation, and then did in vivo optical and then MR. So here again is this confocal microscopy. These animals are living. So here's the pancreas of this animal. These are the targeted nanoparticles. We also had something that would illuminate or highlight the blood vessels so we can look at the blood vessels that are there. And then this is one of the caveats of a mouse system, is that there's multiple tumors in here because every cell in the pancreas is getting the mutations needed to start a tumor. But what we can see and keep track of is individual cells that are going through the pancreas and migrating. So doing this, we can actually start looking at drugs that would help us to inhibit that process. We could also do MRI, so this is fluorescence molecular tomography, this is an optical approach, but this is MR, so this is before we injected the nanoparticle, and then this is after. This is a T2 star weighted scan, and you'll know this after we get done with imaging, so don't pay attention to that now. But this helps us localize it. So the lower signal is better. That means more is accumulated in there. And if you wanted to do therapy, you'd use those particles to heat up. You'd hit them with a radio frequency that would cause them to heat and then start to kill the cells. And then here's just, again, the data, the quantitation of this. But we could do biodistributions again, or pharmacokinetics. So here's the blood concentration. So this is the blood half-life of these particles. It's a very long half-life. It's a nanoparticle. It's 40 nanometers. Here's the tumor accumulation after that time, and then tumor accumulation by volume of the tumor. And then this is called a biodistribution. And so after we're done with the animals, we sacrifice them, we weigh each of the tissues, and we quantitate the percent injected dose per gram of tissue. So here it is in the tumor. There's a lot of this in the tumor. Not as much with the control nanoparticle. So this is a particle that does not have a peptide, but you can still see that you can get some in there due to the EPR effect. So it's not like you're not getting targeting, it's just you're getting more targeting into there. The lung, the heart, the skin, the muscle, spleen, and kidney, again, and liver, because those are the RES organs. They have to go. And then look at the normal pancreas. So you can see that with the targeting, we're getting less in the normal pancreas than if we just use the negative nanoparticle or the non-targeted nanoparticle, and more is in the tumor. So the ratio of tumor to normal is much higher with the targeted, which means we would be able to change the efficacy to toxicity ratio to the good. From there, we've looked at other proteins. This is a spark secreted protein, acidic and rich in cysteine. This is a protein that binds to that binds to the extracellular matrix. This is a protein that's very important in all invasive cancers. We have made through phage screening new spark ligands. We've been able to make spark targeted agents. Here's a different kind of nanoparticle that's coated with a carboxymethyl anhydride. And we've been able to show that it measures more aggressive prostate tumors. So the more spark there is, the more this is. So this is an example of imaging with these nanoparticles. But we're doing imaging here to simulate drug delivery so we can quantitate how much drug would go in there. So the fluorophores are either hydrophilic or hydrophobic depending on what properties we're choosing, that we can load into the drug or into the liposomes or whatever particle that we're choosing. And in this way we can work out the loading parameters, and we can also work out how much of that drug we've actually loaded. So when we choose something that doesn't allow us to visualize it, we've worked out the conditions on something that allows us to track it. And then we can track it both in imaging and also by doing the biodistributions. So that in a nutshell is the targeting lecture. And if you have any questions, please start a forum on Piazza.